Hi everybody, welcome to Learn Medics. Today we are going to deal with apoptosis. In this module, we are going to deal with the basic mechanisms of apoptosis and cellular changes that occurs in an apoptotic cell. Initially, we are going to deal with the basic concepts of apoptosis. Apoptosis is also called programmed cell death. It gets its name by the cell activates their own machinery and their own enzymes to destroy cell. It is in contrast with the necrosis because the membrane remains intact. Ultimately, the apoptosis occurred cell consumed by phagocytes. That is, example, cells like macrophages consume it by phagocytes and destroy the cell. It is a non-inflammatory process, so that it process no damage to adjacent cells. Single cell undergo apoptosis and destroy, and it does not possess any damage to adjacent cell. So it is in programmed and orderly manner, that is in contrast to necrosis. And also for this programmed process, the ATP is required for this programmed process. So it is an active process. We know that some stimuli cause both apoptosis and necrosis. For example, historically, it was thought that myocardial infarction forms of cell death was initiated by necrosis, but later studies proven that initial response is by apoptosis, that the cell adapt to the infraction by apoptosis way, which utilize the ATP and it does not possess any inflammation. But later apoptosis, that is, if apoptosis get fails, it leads to the necrosis, which is due to the ATP depletion and failure of apoptosis. Basic causes of apoptosis. Initially, it is a normal part of embryogenesis. There are many structures that present in embryo which does not persist to the adulthood or in childhood. So those structures undergo apoptosis, and cell dies itself in an orderly and programmed way. Second one is the hormonal withdrawal. It occurs in tissues that depend on hormone. For example, in the case of endometrium, in the menstrual cycle, we know that there is an progesterone and estrogen cycle occur in menstrual period. So when the progesterone withdrawal occur, the endometrium falls off. So the falls off means it goes on a programmed way by the apoptosis way. It occurs in the hormonal dependent cells. Next one is immune cells. We know that to attack many pathogens, immune cells produce rapidly. So after the pathogens, the immune cells that more produce should be died. So it is died due to the apoptosis way. For example, there is a rapid production T cells and B cells in thymus. So it should be normalized after the inflammation or the attack by the pathogen go. So it is done by the apoptosis way. Second, in DNA damaged cells, the DNA damaged cell should be undergo apoptosis and it should be die in an orderly manner. And there is another type, means abnormal cells. We know that any mutation that occur in a cell leads to the tumors and cancers. So the cells need to be go in a apoptotic way if any mutation occur. So it is, if that is not gone in an orderly or manner, it leads to cancers or tumors. So it is a basic for all the abnormal cells that should be undergo apoptosis. Otherwise, the other thing is the infected cells, especially the viral infected cells, also should be undergo apoptosis. So these are the main causes of apoptosis occur in a cell. Till now, we are discussed with the basic concepts of apoptosis, the causes of apoptosis. So how this apoptosis occur? How a cell process the programmed cell death. So initially, if a cell needs to be apoptosis, the main thing we need in the cell is the caspase enzyme. The caspase enzyme is the main thing that we need in a cell for the apoptosis. It is inactive in cytosol, but if the activation leads to apoptosis. So the caspase enzyme, initially it is inactive in cytosol. It is present in a cell but is inactive in form. But the later responses or the stimulus leads to the apoptosis. Activation of caspase enzyme leads to the apoptosis. The two basic ways or the pathways for activation of a caspase enzyme is by the first one is the intrinsic way or the mitochondrial way, which is initiated by the mitochondria. So mitochondria situated intrinsic to the cell, inside the cell, so it is called intrinsic way or the mitochondrial way pathway. Second one is the extrinsic, extrinsic or the membrane death receptors, which is activated by the death receptors that present in the membrane. These are the two ways that the apoptosis, the caspase enzyme activated and leads to apoptosis. So, how this intrinsic or mitochondrial pathway and the extrinsic pathway are activated? So, we are going to discuss with that. First one is the intrinsic pathway of activation of caspase enzyme, which is initiated via opening of mitochondrial pores in the mitochondrial membrane, which leads to the release of cytochrome C to the cytosol, which binds to APFA1 or called apoptotic activating factor. The ability of cytochrome C to attach to or to bind to APF1 is controlled by the BCL2 families of proteins. There are two types of BCL2 families. First one is the anti-apoptotic, means it restrict the apoptotic mechanism. Otherwise, in the pro-apoptotic, which leads to the proteins leads to the initiation of the intrinsic pathway and leads to the apoptotic. So the balance between those, that is anti-apoptotic and pro-apoptotic proteins present in the cytosol, leads to the whether the cell determines whether the cell undergo apoptotic or not. So this is the basic requirement, that is the balance between pro-apoptotic and anti-apoptotic proteins. So we are going to deal with the what kinds of proteins are the anti-apoptotic, which inhibit the apoptosis, or what are the pro-apoptotic, which is promotes the apoptosis. So the anti-apoptotic proteins are the BCL2, BCLXL, and MCL1, which is found in mitochondria, which keep membrane intact. Means the we already discussed that the pores of mitochondria needs to be open. So these type of proteins, that is BCL2, BCLXL, MCL1, found in mitochondria, keep the mitochondrial membrane intact. So it prevents the cytochrome C to leak. Thus the apoptosis is inhibited. So the pores does not open. So the cytochrome C fail to leak to the cytosol, and it is failed to bind with the APF, that is activating apoptotic factor. So the anti-apoptotic inhibit the apoptosis. Then how the apoptosis occur? It is by the pro-apoptotic uh, proteins, which is high yield. The backs and back proteins are the pro-apoptotic proteins which bind to anti-apoptotic proteins. Like these backs and back bind to BCL2 type of or BCLXL type of or MCL1 type of proteins which inhibit them, and it leads to the opens of mitochondria. Open pores in mitochondria leads to leakage of cytochrome C to the cytosol that initially promotes the apoptosis. The triggers are the first one is the withdrawal of growth factor, that is in a decrease in hormone, like hormone sensitive tissues like uterus. Any decrease in hormone leads to triggers for the activation of intrinsic pathway. Any decrease in cytokines for immune cells also leads to the triggers or the activation of intrinsic pathway or the embryogenesis, which all goes under the withdrawal of growth factor. Withdrawal of growth factor leads to the activation of intrinsic pathway. How this activated? Via the mitochondrial opening, the cytochrome C, mitochondrial pore opening, cytochrome C leak into the cytosol and binds to the APFA1, and thus the APF1 promotes the apoptosis. Thus, the withdrawal of growth factor leads to the apoptosis. And second was the DNA damage, which I already mentioned. From radiation to chemotherapy, we have DNA damage, and CYP53, that is cytochrome P53, can activate backs and back, and it leads to the trigger the intrinsic pathway. The third one is the abnormal proteins, which is caused by heat, hypoxia, and low glucose. These are the triggers of intrinsic pathway. Extrinsic pathway. As I already mentioned, that extrinsic pathway is activated by the death receptors present in the cell membrane. So it is extrinsic, outside, externally. So the death receptor found on surface of cells leads to apoptosis, which is a part of tumor necrosis factor or TNF proteins, and it span plasma membrane and spread into the cytoplasm from outside. The tumor necrosis factor spans into when activation of death receptors. So it spreads into the plasma membrane, and invert to the cytoplasm from outside. The FAS ligand, or well described with FAS ligand or CD95, is the well described death receptor found on many cells, which leads to binds to FAS ligand, which triggers sequence that leads to activation of caspase enzyme. Triggers for activation of the extrinsic pathway. The first one is the CD8 T cells. When a virus infects a cell, the cell activated by presentation of viral antigens. We know that for the adaptive immune system, the antigen should be presented. That is called antigen presentation should be occurred to activation of the adaptive immune system. That is B cells and T cells should be activated. So it need an viral antigen presentation, and the MHC1 binds with the CD8 T cells and release the perforin and Grans enzyme. The perforin and Grans enzyme leads to the activation of caspase enzyme, which binds to the FAS ligand, FAS CD95 on surface on cells that activates the caspases. Initially, I mentioned about the FAS which found on the surface of death receptors. So the FAS ligand occur and which activate the caspase enzyme. And second one is the thymic medulla. T cells that bind to self antigens die in a negative selection. It is a negative selection. So death occurs via extrinsic pathway of apoptosis, and FAS ligand interaction between T cells and thymus cells leads to apoptosis. So the negative selection of T cells that bind to self antigens leads to the FAS ligand interaction between T cells and thymus cells leads to apoptosis. That occurs in the thymic medulla. Till now, we discussed about the basic concept of apoptosis and the extrinsic and intrinsic pathways for activation of caspase enzyme that leads to the apoptosis. So, we are going to deal with the cellular changes occurs in a cell that undergoing apoptosis. First one is the cell shrinkage, which is contrast with the necrosis. But in necrosis, the cell is swelled, and the chromatin condensation, which I already mentioned in my cell injury video, that is the pyrokinesis. Chromatin condensation means pyrokinesis, and membrane blubbing, which is also I discussed in blebs occur in the cell, which I already discussed in cell injury video. The apoptosis bodies, the cell first shrinked and the chromatin condensed by pyrokinesis, and the membrane gets blubbed. The membrane blubbed apoptotic bodies formed by cell explodes and fragmented, which contain the organelle and ligand for phagocyte receptors. So the cell that undergo phagocytes, and initially the cell die. This is how the apoptosis way of cell death. Last slide of this mojo is DNA laddering. It is a laboratory technique for identity of apoptotic cells. The apoptosis caspase cleave DNA at specific regions, for example 180 and 185 kbp. It forms a ladder, which I shown in the figure. It forms a ladder fragments of DNA on cell electrophoresis, which is contrast to necrosis, because it forms only random fragments, which is a laboratory technique to identify the apoptotic cells by the DNA laddering. So I am ending my video. Thank you everyone for listening for me. Thank you and have a great day.